Uh, I'm going to approach the topic of the session and of the conference from a slightly different perspective. I've been interested for many years now in understanding the brain as a complex system, as a complex network, most recently. And so the question arises, how can that perspective inform our understanding of cognition? And I have no good answer to that question. I have a few ideas and I will share those with you. And perhaps we have a discussion at the end and we can untangle some of these thoughts. If you've been in cognitive neuroscience or in systems neuroscience or neuroscience in general, networks have been on the rise over the last 10 years, really. And there's almost no empirical system, species, recording methodology that has not been subjected to some level of network analysis and modeling, going all the way from nematode, C. elegans, classic model organism, to the mouse, to the non-human primate, and of course the human brain with neuroimaging methodology. And this notion of brain networks has taken on a somewhat different meaning, I think, than it had been taken on in the past from the era of artificial neural network research. It's a slightly different concept here. It has more to do with complex systems and topology and graphs, as was mentioned at the end of the last presentation. Why this rise in network approaches? I think it has to do with the fact that our recording methodologies, our data gathering abilities in neuroscience have grown exponentially. We are now inundated with information on molecular networks, on cellular circuit, whole brain networks, and also social networks and behavior. And increasingly, those data are being conceptualized as relational data, data that have to do with relations among elements in a complex system, and are being analyzed and modeled with network science methodology. For the most part of my talk today, I'll stay in the middle of this field in the realm of connectome networks, particularly as constructed from human neuroimaging data. When I got started in neuroscience, the knowledge of the human brain was quite limited still. And when it came to anatomy, it was really limited to postmodern dissection, histology, obviously not ideal from the perspective of understanding anatomical underpinnings of cognition, because the specimen in this picture is unfortunately long deceased. It's a photograph of a dissected brain. You can see the white matter architecture, the white matter tracts going through the interior portion of the cerebral cortex and linking distant brain regions with each other. Only quite recently have we been able to image and measure and reconstruct anatomy in living people, in living organisms, and living humans using diffusion imaging and tractography. While this technology has many technical difficulties still to overcome, it is ultimately a computational modeling approach and inference of connectivity as we understand it from diffusion signaling. It has given us a more and more refined picture of how connections are laid out in the human brain, in particular in the cerebral cortex. And in my earlier work with Patrick Hagman, we built some of the very first connectivity maps, connectivity matrices, one of them shown here. Essentially a lookup table of what's connected to what in the human cerebral cortex, with the right and left hemisphere indicated here. A few features of such a matrix are apparent. It's a sparse matrix. It has some symmetry to it, and there's varying strength of connections shown here in a color map. So connectivity between brain regions, between populations of neurons, widely distributed across the cortex. And this is one of the snapshots that we generated early on in our studies. This field of connectomics has really exploded, and there are dozens, hundreds of labs really in the world doing this kind of type of work now. I think we'll hear a lot more about this from Lucina's talk later on. In our first investigation of how the cerebral cortex is connected, a few features stood out, and they have broadly been reproduced over and over again in different cohorts and in different imaging centers across the planet, really. Each patch of cortex has its own unique connectivity fingerprint. By that, we mean a set of inputs and outputs that are unique to that part of the brain. In fact, in a network, who you're connected to, who you're talking with, can define very dramatically the functional capacities that you have. So we view these connectivity fingerprints as indications of functional specialization. Some brain regions have a lot more connections than others. There's a broad, perhaps log normal degree distribution across the brain. We're finding small world properties such as high clustering, high propensity of nearby regions to be connected, and overall short path length information can be distributed fairly liberally, fairly easily across the entire large network. We have modular architecture. The modules are interlinked by important regions that link modules to each other, and we call them formally hubs. And these hub regions are interconnected into a structural core. That last feature is of particular interest, perhaps, for the current discussion. Why? Because it might form the infrastructure, the underlying highway system that enables distant brain regions in different modules to talk to each other very quickly and very efficiently. Let me explain to you what I mean by that. This is a diagram that is supposed to illustrate the so-called rich club feature, rich club organization of networks. On the left is a simple network, a schematic organized into 4 modules interlinked by regions that link the modules to each other, shown here in black, so-called hubs. If we add a few more connections among these hubs, we now form an interesting feature that sort of hovers on top of this modular architecture and connects the modules to each other, as well as those highly connected hubs to each other very quickly, very efficiently. That's a feature that in social networks is called rich club. We see it in human brains, but also in brains of other species. And it is a non-trivial feature of network organization. It doesn't have to be that. It's actually quite costly to implement, and seems to, in our view at least, seems to go very well with ideas of global communication in the brain, information sharing, creating short paths that link highly processed information to each other, perhaps something like a global workspace. The rich club organization seems to be quite well suited for that type of information processing architecture. Working with Martin Mannenheuvel, we set out to look for a rich club organization in the human brain. We found it on our first try. Shown here, illustrated as this increase in density of connections for high degree nodes interconnecting with each other compared to a random null model. The rich club itself, the membership of it includes regions like cingulate cortex, posterior parietal cortex, the insular and middle temporal regions as well, all well known in cognitive studies, of course, as carrying and processing highly specialized multimodal information. It turns out these regions are interconnected amongst each other more densely than expected by chance. That is the hallmark of rich club organization. From a network science perspective, if you want to go from any place to any other place in the brain and plot a path along which information might be able to travel, it turns out that the rich club soaks up much of that traffic. 9/10 of the communication paths across the brain touch upon some aspect of it, even though the rich club itself is something like 10% of the infrastructure in total. Another connotation that I can't pursue out of time is that damage to rich club neurons, rich club regions, rich club connections, network science would predict has a disproportionately large effect on network integrity, on efficiency and communication processes, perhaps integration. And it turns out that there is building clinical evidence that rich club damage is indeed particularly impactful in terms of negative impact on human cognition if rich club damage does occur. We've done a lot of our work in humans, but I want to give a quick nod here to model organisms. After all, they're very important in laboratory studies. And connectomics, of course, has taken its origin really in the nematode C. elegans and has really impacted all of these different model organism investigations over the last decade or so. We in our lab have worked on reconstructions of the Drosophila brain, the rodent, as well as the non-human primate, of course, humans as well. And it turns out that many of the features that we found historically, actually, first in human brains, modularity, rich club organization, important hub regions, they are shared across these different species. We find modular rich club organization in species as simple as the nematode C. elegans or the invertebrate Drosophila brain. Historically, again, working with my late colleague Olaf back in the day, we started our investigations in macaque cortex. I want to briefly say a few words about these model organisms because in model organisms, the methods that we have for reconstructing connectivity and especially anatomical graphs are much more sophisticated, much more precise, much more ground truthy if you want. And in fact, the field had at its origin studies carried out over decades, really, in histological track tracing in the non-human primate. And Wolf and I started our work back in the day, mining the very few data sets available to us, very sparse, very precious data at the time. Back in 2005, 2004, 2005, we had no such map for the human brain, and it's worth reflecting for a moment how explosive that growth in knowledge has been over the last 15 years of connectomics. We had no corresponding map for the human brain at all. We are now awash in data. Thousands of such maps have been created for all sorts of individuals across all sorts of groups and settings and different imaging centers. And we have a deluge of data that we now almost don't know what to do with. Working with my colleague Larry Swanson at USC, we have embarked on a series of studies of different subdivisions of the rat cortex. Once again, advantage, histological track tracing allows us to see connections at much finer detail than we could with tractographic reconstruction. And Larry builds these fascinating maps of directed connectivity for different brain regions of the rat cortex shown here. We have extended these studies into basal ganglia, hypothalamus, midbrain, thalamus, etc., and are planning to go into the cerebellum and hindbrain very quickly in the next iteration of this study. And we're finding different features of network organization in these different structures, different anatomical subdivisions. The cortex shown here, it looks very different to basal ganglia or to the hypothalamus for that matter in terms of the connectivity topology that we actually see. And we do think that those differences in topology reflect differences in information processing strategies, demands that these structures subserve, and connectomics is beginning to get us closer to understanding some of these primitives, some of these elementary patterns of connectivity that go into those different competencies. One thing that stood out for me over the years is how similar brain organization at the anatomical level looks across different species. And working with Ed Bullmore, we came up many years ago now with a way of explaining why that might be so. And it's worth pursuing for a minute because it does relate in the end to our notion of what cognition is and what it's for and where it comes from. So in our hands and the hands of many other practitioners, brain networks have what we call complex topology. What do we mean by that? We mean there's a mixture of local features such as modules, which are densely connected clusters, with some long distance connections thrown in that are highly non-random, linking different brain systems to each other, different modules, and are probably critical for integration across the brain. So this complex topology is actually also, we can think of it as an expression of a simultaneous trade-off of two different selectional forces that may have forced the brain into this regime. One is preservation of wiring length or volume or cost in general. The brain cannot be infinitely costly, obviously. An organism cannot use more ATP than it can actually produce. That's a lethal, that's just not survivable. So brain architecture is subject to strict constraints of cost in terms of material, volume, but also energy. Brain wants to be as cheap as possible, is another way of putting it. It also wants to be moving information around as quickly, as efficiently, and as adaptively as possible. You know, Randy's talk gave a beautiful example of how that might be implemented. These two constraints are mutually incompatible. If the brain wants to be really cheap, all connections are short, and we're having a real problem moving information from the back to the front of the brain. If the brain wants to be infinitely fast in moving information around, it gets to be randomly connected. That's not possible to wire with given volume and cost constraints, and also very bad because we're losing functional specialization as a result. This trade-off in the middle is sort of like almost like an engineering context, a sort of multi-objective optimization that's been behind this. And we feel that the similarity of architectures we find across very different species that live in very different ecosystems at very different sizes, etc., reflect somehow this universal need to trade off cost with efficiency. And that now leads me to cognition. I feel like there's a real point of departure here with artificial neural networks, which generally do not have those constraints. The architectures that, DeepMind can realize in an AI system, there's no constraint really of wiring minimization. There's no constraint of ATP consumption or energy, and information can flow in any which way, and there's not much dynamics actually quite often. So I wonder whether the space we find ourselves in terms of our actual connectivity that our brain is endowed with provides us, it's sort of like a very narrow slice of the cognitive space that's available in principle to neural network architectures in general. We can maybe unpack that later. Last point I want to make is, so I've talked about anatomy a lot here, and I think Lucina will talk a lot more about dynamics and flexibility and functional connectivity, but just to give you a brief taste of how these two worlds relate to each other, we have a sort of static anatomical skeleton that we can measure with connectomics reasonably well, and then we have on top of that swirl of activity that goes on, in this case, a movie of a person lying in a scanner doing nothing in particular, the so-called resting state. And so the question that my lab has been working on for many years now is, how do we get from something that is relatively static and sparsely connected to something which is as dynamic and flexible, either in rest or, as we'll see later, I guess with Lucina's talk also, during task, that has these varying patterns of activity and connectivity over time. One way that we can address that is by measuring functional connectivity. We do this by taking a movie like the one we've just shown you and cutting it up into frames, cross-correlating the time courses that we get from those bolt signals. And that gives us a fairly reproducible picture of what we now call resting state networks, which are really network modules of densely connected, coherently active and coactive brain regions that sort of go up and down together over time during rest. Many of these networks now have names that are dear and that are very dear to us, like default mode, the limbic system, the saliency system, the visual system, etc., which have connotations with respect to task domains. We see these systems pop into high gear and becoming coactive also during task settings. So rest and task, in my view at least, are continuum. Rest is something which is task-free. Tasks are more constrained subsets of what the brain can realize as it undergoes resting fluctuations. These patterns of resting state networks are very reproducible across imaging centers and cohorts. That's why we have these maps now that are very canonical, that we can apply to brains across the world, really in different context, measured in very different contexts from very different people. Everybody on this webinar has a default mode network that's roughly laid out in the same way as we see it here in this bright red patchy image at the bottom. This has given us in cognitive neuroscience sort of a new way of parsing the brain into systems and understanding how these systems become engaged during different cognitive processes during rest, and how they are related to the underlying anatomy. So a couple of words at the end about modeling, where functional connectivity comes from. Resting brain activity, we take it to be spontaneous, unconstrained, at least unconstrained by an external stimulus, spontaneous activity unfolding on top of a relatively static skeleton of anatomy. We can build generative models in the sense that these are models that generate data that are similar to empirical data we can compare. These models can take the shape of coupled differential equation models, sort of a physics modeling approach to the brain, where each node in the network is a neural mass that has certain characteristics of excitation, inhibition, etc. And we can simulate its activity in a computational neuroscience framework, generating synthetic bolt signals in the process. We can then compare to empirically recorded bolt signals shown here on the left. The model is on the right. These two are not perfect, not perfectly correlated. There's been no parameter fitting at all, actually. The model was created many years before the empirical data was collected. Nevertheless, some degree of agreement suggesting that indeed the anatomy can shape functional connectivity in a real macaque brain. On the right, a model, a much simpler model, analytic model, a graph analytic model that we've used in human data, where again, the right part of the diamond here is the prediction from the model. On the left is the empirical functional connectivity from an actual scanner. And the agreement, while not perfect, is significant and suggests to us that indeed functional connectivity, the statistical dependencies between brain regions as they fluctuate over time and during rest in this case, are driven or shaped or constrained at least by underlying anatomical patterns. So a role for the connectome in shaping statistical patterns of activity, coactivity that we can observe with functional brain imaging methodology. A few words at the end about work that is fairly recent in our lab, only last year or two, but I'm very excited about. I want to share with you a little bit. Functional connectivity of the kind I've just shown you is typically estimated over a long period of time, during rest or during task, many time steps, many different TRs, as we call them in the scanning business, many minutes of time that are being coalesced into a single covariance matrix, a single image of what the dynamics look like. But we can actually, it turns out, decompose functional connectivity into blow-by-blow accounts of what the brain is doing. By blow-by-blow, I mean as fine as the single TR resolution of an MR scanner, so about 1 second or so in time resolution. We can do that by unfolding functional connectivity, which is a Pearson correlation typically, into a set of bold activations that on every edge can be reduced to what we call a co-fluctuation amplitude, which is really the product of the two bold signals across time. It turns out that the average of this time series, which is now an edge time series, is exactly equal to what has been classically used in many studies to represent functional connectivity. We simply roll that process back one step and represent what is going on between two brain regions as a time varying account of whether these regions agree or disagree in terms of their activation status. You can think of this as a time series on each edge of the network. When we build a matrix of edges by time from such a data set, first thing, if we take the mean of that matrix that you see here along the y dimension, you could be getting a vector which we can fold into an FC matrix. This is the construct on the left. The functional activity matrix that you see is widely used in our field to represent something about resting dynamics or task dynamics in human brains. So nothing has been added here. It's sort of an organic model of functional connectivity. Nothing's been added that shouldn't be there. No fancy information theory. Nothing's been added. Something has been taken away, which is the averaging step. If we, as you can probably see just by looking at this matrix, there are these very worrying vertical streaks running through it. We were very worried about them because every fMRI researcher is always worried about signal artifact and nuisance variables and heartbeat and yawning and whatnot. Turns out this is none of those things. And these vertical events, as we call them informally, represent high co-fluctuation amplitude, brief bursts of high amplitude co-fluctuations that seem to be engulfing most of the brain. These are found these are unrelated to nuisance or physiological artifacts. We see them really in all individuals. We've seen them also in movie data, where interestingly enough, they are somewhat aligned across subjects and in time, in terms of the time when they occur. And this suggests that there is some relation to stimulus. And they are present in all individuals, as I said here. We see them they can be seen in animal data, and they can also be seen, by the way, in dynamic simulations. I want to show you what these look like in the form of a movie. This is my last item here, and then I'll wrap up in just a minute. What you're seeing here is a totally normal fMRI run from the Human Connectome project that's been unwrapped in time in our new in the new way that we've just been introducing here into edge time series, which you can see slowly accumulating as the run progresses. On top here, you see a top-down view of a brain matrix, 200 nodes by 200 nodes. with classical functional systems indicated by these little squares, default mode, frontoparietal, limbic, you name it. Red means positive co-fluctuation, blue means negative co-fluctuation. And each TR here is a snapshot of how different nodes, different regions of the brain agree or disagree with each other in terms of their activation status. If you take the global mean of the movie in the square at the top, what you get back is exactly functional connectivity. Mathematically, and not an approximation here, that is an exact thing. But as you unwrap it, one thing that strikes us, and we're looking into more and more and more, is that those classic functional systems, such as smarter motor, visual, etc., are sometimes clearly realized in time. They do appear as clearly visible red squares in that movie. We can do statistics, of course. This is not just visual impression, but I'm showing you the movie here just to drive home the point. But the entire functional architecture, the picture that we see from functional connectivity really never is that clear. So functional connectivity in the long term is a superposition of many dynamic states that we now have perhaps a way of disentangling across time. We're very excited about this and have done a lot of work, a lot of ongoing work in the lab and with our collaborators to dig into this more. Last slide, the picture I want to leave you with, from my perspective as a connectivity researcher, goes a little bit like this. I think about the brain in very structural terms. I think this has to do with my training a long time ago as a biochemist, when I was very enamored with protein structure and how much protein structure can tell us about how proteins function. Structural connectivity, the connectome, as we've called it 15 years ago, underpins what goes on dynamically, a level of interaction that I'm very interested in, that we don't see very well with current empirical methodology, is communication dynamics, the flow of signaling events in such a network, which then manifests in terms of output as activations, co-activations, functional connectivity, however you want to call it. This is what we measure with fMRI or classic recording techniques. Once again, this middle ground here, which is very invisible to us at this point, the dynamic flow of signals in the wiring, in those axonal pathways that I've shown you earlier, that's the level I'm more and more interested in my own work. Lastly, a few thoughts about cognition. At least from where I am sitting, cognition does emerge from brain networks. Yes, we can build cognitive architectures in an artificial setting as well. An open question then is how, what are the common features that we can glean, or is there a point of divergence ultimately, where cognition that emerges from brain networks is ultimately different from the kind of cognition we can get from an artificial setting? Features of brain networks that we are interested in so far: modules, hubs, the interconnection at the rich clubs. Many of them are shared across species. There's a common evolutionary history that we share with other species. There's a continuity. Whatever capacities we may possess as humans, of course, there is continuity, to me, of course, with other species, non-human primates, other mammalian species, and also with invertebrates. The evolutionary pressure on networks that they're subjected to really are massive when it comes to issues like cost and efficiency. By cost, I mean wiring volume, ATP usage, energy consumption. Efficiency, I mean how quickly, how efficiently can we move information around. Those two constraints trade off against each other, and the cognitive capacities that emerge from brain networks are subject to the same constraints. So to put it differently, perhaps there's a cognition somewhere, and perhaps it's AI, that isn't subject to these cost and efficiency constraints, and therefore much more powerful and very different, very alien to what we are used to as humans. Finally, this is, sorry, I'm going over by like a minute or two here. Finally, segue into Elisina's talk. There's a growing consensus in our field that cognition depends on reconfiguring that pattern of dynamic interactions that you've seen in that movie in an adaptive way, in a way that is flexible and adaptive. And I think Lucina will have a lot more to say about this. Finally, there's a journal that I'm running that deals with network neuroscience, in case you're interested. So thank you very much for that.
Thank you so much, Olaf, for this tour de force of brain connectivity. We're running a little bit, maybe a little bit under time, so I'm I'm just going to pick a couple of questions that I think will clarify your talk. A couple of people actually have asked clarification about the time scale of this functional connectivity. And especially Peter has asked whether it is related to the idea of the 50 millisecond cognitive cycle.
Yeah, so with what I showed last, the finest temporal resolution we can get to is one image, and that's roughly 700, 800 milliseconds in the case of the HCP acquisition. So we can't see beyond that with MRI. However, of course, we can look at this with electrophysiological EEG, MEG, or invasive recording methodologies, and that's something that we have begun to do. There's clear non-stationarities there. There are interesting brain-wide events like ripples and waves that may relate to the events that I've shown you in the MRI picture. MRI is just a very widely used technique, and until now it's largely been used in a sort of as a summary of a lot of time compressed into one image or one matrix. And I'm very excited that we can now sort of, unwrap that a little bit more and look at the dynamics as they unfold on a finer time scale. Not perfect, not as fast as indicated in the question, but down to about 1 second.
There are actually two great questions that I will not have the time to actually ask right now, but I invite you to answer. One is from Paul Rosenblum, and the other one from Keith Lambert. I would like to ask a question that was posed earlier by Pat Langley, because I think would be a nice segue to Lucina's talk. And it's whether the different individual differences in these connectomes could be related to individual differences in function and cognition.
Yeah, I mean, that's Lucina, I leave this one for you because you will address it, I'm sure. And the quick answer is yes. A lot of activity goes into a lot of research is going into understanding those relationships better. For me, a limiting factor still is that our measurement techniques aren't as good as we would like them to be. And so we can see some of these relationships, but they're a little bit indistinct still. It's early days. It's only been about 10 years that we have been able to do this, which in science is a very short period of time. So I think, Lucina, you will address all this in much greater depth than I can possibly do in this question here.